In a typical animal cell, the cell or the plasma membrane defines the outermost boundary of the cytoplasm. Thus, in an animal cell, the cell membrane maintains its integrity and shape. The cell membrane is mainly made up of lipid layers, that is, bilayer, in which the protein molecules are embedded. The membrane is selectively permeable in nature and thus regulates the flow of materials into and out of cells. The part of the protoplasm present between the cell membrane and the nucleus is called cytoplasm. The cytoplasm of an animal cell is a transparent, viscous, colorless fluid. In the cytoplasm, various organelles are present and each is concerned with some specific function. The cytoskeleton is a network of fibers throughout the cytoplasm of a cell. It is the structural framework of a cell that helps in maintaining its shape and facilitates cellular motility. The cytoskeleton is an organized network of three primary protein filaments: microtubules, microfilaments, and intermediate filaments. All the activities occurring in a cell require energy in the form of adenosine triphosphate or ATP. The mitochondria are a site of ATP formation, and thus these are also known as the powerhouse of a cell. Mitochondria are rod-shaped structures present in eukaryotic cells. These range from 0.5 to 1 micrometers in diameter. Their number varies from one cell to another. A mitochondrion is a cytoplasmic organelle with double membranes which surround the fluid-filled space or mitochondrial matrix. The outer membrane allows low molecular weight molecules to freely diffuse from one side of the membrane to the other. On the other hand, the inner membrane of the mitochondria is highly convoluted, forming a series of infoldings called cristae. Cristae provide an increased surface area for all the enzymatic reactions that occur in the mitochondrial matrix. The mitochondrial matrix is a dense, gel-like proteinaceous material. The mitochondria are the site of cellular respiration. Glucose and oxygen react to give carbon dioxide and water, and energy is released in the form of ATP. Ribosomes are small, dense, rounded, and granular particles. They either occur freely in the cytoplasm or remain attached to the endoplasmic reticulum and the nuclear membrane. Protein synthesis takes place in ribosomes. The endoplasmic reticulum is an extensive and a complex network of interconnecting membrane systems, which join the nuclear membrane at one end. Functionally and structurally, two types of endoplasmic reticulum have been recognized. These are granular or rough endoplasmic reticulum, agranular or smooth endoplasmic reticulum. The endoplasmic reticulum is composed of three different types of structures: cisternae, vesicles, and tubules. The endoplasmic reticulum acts as a supportive framework for a cell. Some of the important functions performed by the endoplasmic reticulum involve synthesis and transport of proteins and fats. The Golgi complex is about 1 to 3 micrometers in length and is scattered throughout the cytoplasm, mainly near the nucleus. The Golgi complex, like the endoplasmic reticulum, consists of 3 main parts: cisternae, tubules, and vesicles. The Golgi complex is mainly involved in the packaging of materials that have to be either delivered to the intracellular targets or secreted outside a cell. The next organelle that we encounter in an animal cell is a vacuole. In a typical animal cell, the vacuole is not as prominent as in a plant cell, and the cytoplasm occupies almost the entire cell. Animal cells have fewer and smaller vacuoles. The vacuole is enclosed by a single membrane. The vacuole is filled up with a vacuolar sap. The cell sap is composed of water, phenol, salts, etc. The vacuole serves as a site for storage and for depositing the cellular waste. Vacuoles also perform the osmoregulatory function. Lysosomes generally occur in large numbers in secretory cells like those of the liver, spleen, macrophage, etc. Lysosomes are spherical single-membrane bound organelles having diameter that varies from 0.23 microns to 0.50 microns. It encloses a dense stroma containing 40 or more hydrolytic enzymes. The hydrolytic enzymes destroy and digest the foreign substances around them. They also perform intracellular digestion of cell organelles in damaged or injured cells. Hence, lysosomes are also referred to as suicidal bags. A small, clear region without any granules near the nucleus forms a centrosome. One or two tiny tubular structures that develop in the centrosome are called centrioles. Two centrioles initiate and regulate the cell division by forming spindle fibers with the help of asters. The centrosome is surrounded by microtubules. Apart from these cell organelles, small microbodies and granules are also present in an animal cell. Microbodies include peroxisomes, which protect cellular organelles against the toxic effect of hydrogen peroxide. The granules of starch, glycogen, and fats are the reserve food present as small particles in the cytoplasm. Animal cell is thus characterized by the absence of cell wall and chloroplast and presence of centrosome and lysosome. Rest of the organelles are similar in both plant and animal cells.